Okay, sir, I think we can start. So I'll just give a brief introduction about you and then you can carry over. Very good morning to all. So welcome to the penultimate day of this 10-day workshop. It's a pleasure to have Dr. Arijit Sarkar from High Point University in North Carolina, USA. So I'm pleased to introduce today's speaker. Dr. Arijit is an expert on drug design, computational chemistry, and molecular modeling. He is specifically interested in well-known and ancient diseases caused by bacteria and addressing the problems of antibiotic resistance. Sarkar has been teaching and mentoring students since 2003 and has conducted research on 3 separate continents over the past 16 years. He has a bachelor's degree from Devi Ahilya University and a master's degree from Rajiv Gandhi Technical University in India. After receiving his PhD from Virginia Commonwealth University in 2010, he was a postdoctoral researcher at University of Dundee, Scotland, before returning to VCU School of Pharmacy as a postdoctoral fellow. He has identified potential drugs against multiple diseases, including cancer, infections by bacteria and viruses. He is also a regular participant at national and international professional conferences and a reviewer for several scientific journals. In 2014, he won the International Society of Matrix Biology Young Scientist Award. His specialties include computer-aided drug discovery design, structure-based drug design, ligand-based drug design, quantitative structure-activity relationships, molecular interaction analysis, scientific software development, structural bioinformatics, molecular transport systems, statistical modeling, predictive model building, and validation. And I welcome you, sir. So you have to stay.
Thank you, Dr. Venkat Subramaniam. I really appreciate the very kind introduction. Can I share my screen now? Is that okay?
Yeah. Please go ahead.
Wonderful. Okay, let's go ahead and share that just to make sure that everybody can see what's going on. Are you still seeing me, isn't it?
Yeah, I can see your screen as well.
You can see my screen?
Yeah.
Okay, good. Just wanted to make sure. Right, so I have, if you will notice, I've actually changed my presentation just a little bit. Excuse me for 1 second. I'll try to remove this hide thumbnail. Oops. How can I remove this? Let's put it in the corner right over there. Sorry, it was covering up part of my screen. So you may have noticed that the title that I have at present is slightly different compared to what was advertised. I will be talking about virtual screening, of course, which is the major focus of my discussion today. But I decided to change the title a little bit because I wanted to give a bit of a demonstration of practical approaches to virtual screening using available data in a manner so that students who are watching this can actually learn and understand that there is more to virtual screening than three-dimensional docking, scoring, things of that sort. I will be talking about docking and scoring as well, of course. So let me begin by introducing my lab's focus. Essentially, my lab is working on antimicrobial drug discovery. We're trying to find novel antimicrobials and adjuvants. So the definition of an adjuvant is a chemical which synergizes with known antimicrobials. As you can imagine, both of these can be used as potential therapeutics in the future, although that's quite far downstream. I'm not suggesting that we're going to get a drug in the market within the next few years or anything. That's not the case. We do mainly very early preclinical drug discovery. So another thing that my group focuses on is novel drug target discovery. We will be talking a little bit about this as well. Our group is, as was said in the introduction, we are experts in computational tools, but we also employ some simple in vitro and some very simple microbiology techniques in order to validate the theoretical work that we typically do. And you will see across my presentation that this actually turns out to be extremely useful. So, like I said, I wanted these students to understand a little bit more about virtual screening as a process and as a thought process more than just a set of techniques. So I've made my lecture into a bit of a into a learning lecture, a teaching lecture, rather than just a research lecture. So we have some learning objectives today. So we will try to understand some important problems in virtual screening, particularly in reference to antimicrobial resistance. Specifically, these problems are the chemical space problem. So this is a fancy term, chemical space. What it means is, what are the properties of potential antimicrobials? This is a major problem today, and I'll introduce you to this problem, and I'll tell you certain steps that I have been taking in order to address this problem. The second thing that we're going to talk about is the screen identification problem. Virtual screening is not as simple as picking up a docking program and a target and identifying a bunch of chemicals that dock into a pocket. That is just one part of the screen identification problem. You have to validate these screens. You also have to think about what sort of screens you want to run. Do you really want to run a docking screen? Are there any easier options in order to generate preliminary data which might actually give you a publication or a grant? These are the types of things that you have to think about. Then there's the target validation problem. Do you really want to run a screen against virtually any target that you come across? Probably not, which means there must be some distinction between the good targets that you should be working on and other targets which you may want to avoid. How exactly do you identify these targets for a virtual screen? So as you can see, we're talking about more than just docking and scoring. We're going to talk about a lot of different things and creative ways in which you can approach a screening problem. We will do all of this by looking at live or past drug discovery projects. A bunch of the data that I will show you are published, and I'll have the references for those publications in my slides, so you can just look those up and read them if you like. But also there are some pieces of data which are brand new. They are as yet unpublished, and I'm still working on them. So it should be an interesting first disclosure, I might say, at this time. Okay, so let's begin by talking about virtual screening and a good resource for you to study virtual screening, particularly about docking and scoring. So I'm sure most universities, probably all universities in India that have any interest whatsoever in drug discovery are familiar with this book, Berger's Medicinal Chemistry. So just FYI, the new version, the new edition of Berger's Medicinal Chemistry is due to be released in April. And I and a few co-workers, including my PhD boss, have written a book chapter on docking, scoring, and virtual screening in drug discovery. And if you are trying to pick up the theory and practice of docking and scoring, I highly recommend that you take a look at this upcoming book chapter. I think you might find it extremely interesting and extremely informative. It suffices to say that it's about 900 citations and about 100 pages in print. So good luck reading. I will not be getting into the theory of docking, scoring, and virtual screening. That is left up to you to pick up. We'll be talking about more interesting things, in my humble opinion. Right. So let's begin by discussing antimicrobial resistance, which today is a major problem. If you look at healthcare historically, what you will find is that when we discovered the first antimicrobials in the early 20th century, this discovery enabled us to really advance our healthcare quite a bit. For example, it enabled us to perform surgeries, organ transplants, drugs against arthritis, drugs against cancer, etc., etc. All of these things and more have been enabled simply because we have antimicrobials at hand that will allow us to fight off pathogens. Now, that means we're using antibiotics, antimicrobials. And nothing is more true than this one phrase, which I have picked up from Jurassic Park. You remember the movie Jurassic Park, I'm sure. I don't know how many of you have actually read the book by Michael Crichton, but Ian Malcolm, one of the characters in the book, says, "Life will find a way." This is meant to be for dinosaurs, but this is actually way more true for antimicrobial resistance, for pathogens, for bacterial, fungal, and other pathogens. So as we started to use antimicrobials, these pathogens actually started to demonstrate signs of resistance in the clinic. So antimicrobials, antibiotics that used to work perfectly fine, a good example would be methicillin. It was a very good antimicrobial when it first came out. However, it can't be used anymore. Why? Because most of the bugs today, most of the pathogens today are actually resistant to methicillin. Why? Because we used it in the clinic. Some strains which were able to survive started to proliferate, in turn becoming resistant. So now this has become a really big problem. In particular, because of some less scientific, more financial reasons. So antimicrobial discovery itself is no longer a profitable option for companies. There is very little incentive for companies to invest in antimicrobials anymore. So the resistance coming up, convoluted by the lack of investment, has essentially catapulted the antimicrobial resistance problem in a very big way. In the 1990s and 2000s, barely any antimicrobials were approved for clinical use. For a few years there was zero antimicrobials being approved by the FDA. Small companies emerged which were focused on antimicrobial discovery. Companies like Hewcbds. Unfortunately, Hewcbds closed in 2019. And guess what? Just 1 year before then, they actually came up with an antimicrobial that was active against gram-negative pathogens, which are really, really difficult to treat. Yet it shut shop and went bankrupt. Why? Because people were trying to save these antimicrobials for last resort use. So after investing close to, I want to say about $1 billion in developing that antimicrobial, this company earned only about less than $1 million in year one. Investors pulled their money, the company went bankrupt. And this is happening commonly. Companies like AstraZeneca, GlaxoSmithKline have shut their antimicrobial programs. Why? Because they simply don't see the value in investing all of those things. So making light of the situation, I came up with this cartoon. This is a play on words, resisting resistance. Here's a resistant bug saying resistance is futile. And here's this human being saying vive la résistance, as in long live resistance. it's just making light of a really serious situation, to be perfectly frank. So the need of the hour is novel antimicrobials that work by new methods, new modes of action. And these are extremely difficult to find. Why? Because resistance spreads. And I just want to introduce you to this one publication that really blew my mind when I first read it a few years ago. This was published in 2011 by DeCosta et al., part of Jerry Wright's group in McMaster University. They found that antibiotic resistance is ancient. And what do I mean by ancient? Well, we actually identified antibiotics and resistance in the 20th century. What they did was they went to Fairbanks in Alaska and they dug into 30,000-year-old permafrost. So they had pathogens, they located pathogens which was buried under several feet of ice. Nobody had touched that area for 30,000 years. So they took the bacteria from those reserves, they went ahead and isolated it, they cultured it, they extracted DNA from, genomic DNA from those pathogens, and they tried to identify what resistance-related genes are present. And what they found was that virtually every single resistance pathway gene that we can find today in patients was already present in those bacteria that had never been exposed to antibiotics of a human nature or from which was produced by humans. Why is this the case? The reason is because bacteria and fungi have been fighting chemical wars for eons now. And I'm talking about millions of years, not just a few hundred years or a few thousand years that humans have been on this earth. So the fact that we're using antimicrobials from a fungal origin has made sure already that bacteria have the weapons to fight against those antimicrobials. Again, to reiterate, we need novel antimicrobials that work by methods, modes of action that are completely different from our current set of antimicrobials. So the first question that we must ask ourselves is, how do we solve the chemical space problem? So again, as I had described it earlier, what are the properties of chemicals that are likely to become antimicrobials in the future? That is the question that we must answer at this present time. So let me give you a bit of a background into this problem. Again, we're going back to the 1990s and 2000s, which is when a number of pharmaceutical companies were trying to produce antimicrobials. And like I said earlier, very few antimicrobials were actually identified. So just to give you a few more details, companies have libraries of chemicals of up to about, this is supposed to be 500,000. So 0.5 million chemicals approximately, right? So 500,000 chemicals. They ran these chemicals in 70 high throughput screens. So these are basically screens to identify chemicals which inhibit key enzymes present in pathogens. Can you imagine the number of 96 well plates that we use in order to run these assays? And the amount of manpower and money that went into these projects? Guess what? They identified only 16 chemicals that showed some sort of a promise. And upon optimization, these 16 hits gave them only 5 legitimate leads that could go into clinical development. In other words, on average, approximately 14 high throughput screens of between 0.25 to 0.5 million chemicals would yield only one lead for clinical development. That sort of expense was simply unsustainable. And they ended up culling all of the antimicrobial discovery projects that they had for that reason. And you can read all about it in this publication, for example. There are a number of publications, but I particularly like this one. It comes from GlaxoSmithKline. Right. Another thing that Payne et al. Did was they demonstrated the reason why they were unable to find more chemicals that work. What they found was that if you compare antimicrobials with the screening libraries that they were using, their properties were extremely different. So over here, you see a plot of molecular mass against m log p. So that's your lipophilicity right there, right? Hydrophobic nature. And what you observe is that all of these different colors are antimicrobials, whereas these smaller dark blue rhombi are the typical screening library-like chemicals. And what you observe is that there is a distinct shift in the properties of antimicrobials, which only partially overlap with the typical drug-like chemical. Now, those of you who already know about drug-like chemical, these are also known as chemicals that are compliant with Lipinski's rules of 5. The rules are basically to do with hydrophobicity, size, and the number of hydrogen bond donors and acceptors. Now, I won't get into the details of those, but when I say rule of 5 compliant, I'm talking about the typical tablet that you can take orally and it spreads throughout your body to reach the sites where it needs to act. Antimicrobials are different. Most antimicrobials, such as vancomycin, need to be given intravenously so that they build up a concentration inside blood and they barely reach any other tissues at all. So in effect, what I'm trying to say over here is that antimicrobials were shown to be extremely different in some aspects compared to the typical drug-like chemical that you would find in a big pharmaceutical company's screening library. This was confirmed by one of my all-time favorite publications by O'Shea and Mosier in Journal of Medicinal Chemistry, entitled Physicochemical Properties of Antibacterial Compounds, Implications for Drug Discovery. Again, they demonstrated in a much more comprehensive manner that the properties of antimicrobials, which are the blue and red dots over here, only partially overlap with these gray dots, which are the typical drug-like chemicals. There's only a partial overlap. Same case with polar surface area. Polar surface area versus molecular weight, there is a difference. Relative polar surface area is different as well. And they had a lot more data in there. I'm not going to get into it. In essence, at this point, you should be aware that typical drug-like chemicals, so the typical drugs that you can take a tablet for and it spreads throughout your body, are completely different from the chemicals which actually become antimicrobials. Big pharmaceutical companies had screening libraries, really, really large screening libraries that were focused on chemicals that you could pop into your mouth as a pill and it would spread throughout the body. But these were not necessarily going to be successful as antimicrobials. They had other data in there as well, such as comparing the properties of gram-positive and gram-negative drugs, which were different from, again, the pills that you could pop in your mouth. So what is all of this telling us? Sure, it's a lot of data. There are differences. What is it telling us? If I am to summarize all of this in a single sentence, it's very easy. It literally says we simply do not understand the nature of chemicals that will penetrate bacterial cells. Rule of 5 compliant chemicals, in other words, typical drug-like molecules, may or may not permeate through the bacterial outer envelope to reach the cytoplasm. And we have no way of knowing which chemicals will get inside a pathogen and which one will not get inside a pathogen. This is a big problem, as you can imagine, because what good is it to identify chemicals that are active against an enzyme when that chemical won't even reach the enzyme? This is the summary of the chemical space problem. So again, reiterate the problem. How can we identify chemical space for screening? Before we can begin virtual screening or high throughput screening or even rational drug discovery, we need to understand the type of chemicals which are actually likely to succeed in such screens. So again, what good are chemicals that work against a purified enzyme but fail to reach the enzyme by penetrating the bacterium? This is exactly what happened to the pharmaceutical industry in the 1990s and 2000s, and it is a very well recognized problem at the cutting edge of drug discovery, particularly antimicrobial discovery. So just to summarize all of this in a Venn diagram, if you look at it, here is the set of chemicals that we can actually synthesize effectively. By the way, typical antimicrobials, not so easily synthesized. It's really difficult to handle those, okay? Number 2, here are the chemicals that can penetrate the bacterial cells. And number 3, these are chemicals that can penetrate the human cells. We need to identify some combination of these 3 that will help us identify antimicrobials that will work against the pathogen. So the questions that we must answer are, what are the properties of chemicals that penetrate bacterial cells? How are they different from current collections of drug-like chemicals, that is, chemicals we can successfully and efficiently synthesize? And is there an easy way to identify chemical space where antimicrobials can be found? Note that I double quoted easy because it is not easy at all. Right, so in order to address this problem, we need a study design which has to be based on a large data set of chemicals, which we know can or cannot permeate through the bacterial outer envelope. If you have a well-defined data set, you can compare the 2 types of members, permeators and non-permeators. And this comparison should, in theory, help you understand which chemicals are likely to be permeators or penetrators, which ones are not likely to be penetrators. These need to represent both Rule of 5 compliant and antibacterial-like chemical space. Although it is more important that we deal with Rule of 5 compliant chemical space, essentially because we know that it is difficult to synthesize most antimicrobials. Then we need to ensure that any models we generate are tested very carefully to demonstrate valid and reproducible results. And we finally need to validate our results by testing in appropriate null models and validation techniques. I won't be presenting too much about this last part, but my most recent publication from last year on the chemical space of antimicrobial discovery, it's available online. You can definitely read it. If you need help getting access to it, please just let me know. A good resource for such a large data set of chemicals is actually already available to us. It is known as Hewcbds. I'm not sure if everybody knows what Hewcbds is, but it is a large database which contains chemicals, data on their properties, and on their biological activities. And luckily, this one group had already published something based on the data from Hewcbds. So when I was looking for literature, I actually found the Hewcbds et al. Paper right over here. And they had already done some part of the work to identify a very useful way of identifying penetrators and non-penetrators from that database. And in doing so, they had again demonstrated that there's hardly any difference between penetrators and non-penetrators. They had also a lot of other data, which is extremely interesting, but I won't get into it. But essentially, they had reiterated that the physicochemical properties of chemicals that get inside bacteria and chemicals that cannot get inside bacteria need not necessarily be very different, at least in terms of the chemicals, the physicochemical properties that we typically look at. So I went ahead and used their method and took it one step further, essentially. So this was my publication from last year. It's available. You can read it at any point of time. And what I did was I took the Hewcbds data set and I looked at the data using descriptive statistics. Then I went ahead and built linear and non-linear and even a very simple artificial intelligence model that could make predictions regarding which chemicals would get inside bacteria and which chemicals will not get inside bacteria. And then I went ahead and tested these models for validation and performance. This was essentially covered in this manuscript. I've done further work on this, experimental work in my lab, which I am not presenting today, but it will come out at some point of time soon. Again, the questions I'm trying to answer in this paper are, can we predict permeators or penetrators separate from non-penetrators using the methods or the data set which was devised by Hewcbds? And what are the differences in physicochemical properties? So I went ahead, there was a newer version of Hewcbds with a lot more data available. So I recreated their processes and came up with this data set. In essence, we had a total of roughly about 70,000 chemicals that we know get inside the bacterial cell, and about 9,000 chemicals that fail to get inside the bacterial cell. And I did some detailed analysis of this data set as well. We were able to combine these data to form one set, which is set 3, which would address or identify chemicals that penetrate at least one type of bacterial cell, either gram-positive or gram-negative or both. Set 4 would be able to identify those chemicals that penetrate the gram-positive bacterial cell. Set 5, gram-negative only. And set 6 is the unique set of chemicals that penetrate both gram-positive and gram-negative bacteria. There are some assumptions and weaknesses in this data set. we definitely cannot differentiate between different levels of permeation. So we can only relatively well know that the chemical gets inside or fails to get inside. We don't know, for example, chemical 1 is tenfold better at getting inside compared to chemical 2. That sort of data we do not have. So we're dealing with categorical data only at this time, not continuous data. Second, any chemical that reaches its target is penetrating deep enough. So we're not necessarily saying that these chemicals will get inside the cytoplasm. Could be getting inside the cytoplasm, or it could be acting, say, against a drug target that is present in the outer envelope itself. We do not have clarity on whether or not these chemicals are truly getting inside the cytoplasm, but most of them should, is what we feel. Next, the data is literature derived. So we are already assuming that some of this data is dirty. It's useful, but it is not 100% clean. Unless we generate this sort of data within our own lab, we cannot be 100% certain that we are dealing with good data. The last one, we kept out FDA approved antibiotics because we knew that their properties are significantly different. That would actually be a problem in statistical analysis. Plus, remember that we were also trying to focus significantly more on chemicals that we can easily synthesize and not on chemicals that are, for example, like antibiotics, which are a little bit difficult to synthesize. So remember we talked about drug-like chemicals and antimicrobials, right? So the first question we asked ourselves was, how similar are these chemicals to the typical drug-like chemicals that that might be present in a screening library? So what we found was that a majority of the data set is compliant with rule of 5. In other words, it is drug-like. So you can see it right here. 56% of the chemicals had 0 violations of rules of 5. 23% had only 1 violation. And if you keep the number of violations to 2 or less, which is typically what a lot of screens do, then you are literally covering approximately 56 plus 23 is 79, 89, 91% of all of the data set. So it's a pretty close representation of what a typical screening library in a big pharmaceutical company research and development division might look like. So the next thing we did was we tried to visualize chemical space. When I say visualize chemical space, I'm talking about visually looking at the spread of different properties across multiple dimensions. Or in other words, okay, let me just give you a description and I think it'll become clear. So we use certain software that is freely available, such as KNIME, RDKit, and CDK tools to generate up to 30 properties per chemical. These property generation methods are relatively accurate. So we have a we are able to accept that this is an accurate, at least close representation of the true properties of chemicals. Now, 30 properties is a little difficult to actually visualize, except in a big table, right? Which is not easy to comprehend. So the first thing that we did was we looked at principal component analysis. So principal component analysis is a very simple mathematical method which you can use in order to identify a fewer number of principal components which are almost a substitute for the original properties that you wanted to look at. And we generated, or we found that we could generate up to 10 principal components, and that the first two principal components describe roughly about 67% of the variability within the data set. And if you took all 10 principal components, you could actually describe greater than 95% of the variability within the data set. So let's just take these first two principal components that really cover most of the variability within the data set. So here's principal component 1. Here's principal component 2. These hexagons with different colors mark where your chemicals lie. So this space over here, which is blue in color, is actually roughly about 6,000 or more chemicals per hexagon. It's quite dense. These white hexagons everywhere are 2,000 or less chemicals per hexagon. But it's still a pretty nice spread. And you can actually visualize that most of the chemicals lie within this narrow space right here. And yet these areas over here still have a significant amount of data points. So in other words, what I'm trying to tell you is that this is a very nice collection that covers a lot of different possible chemical properties. So the next thing you could ask yourself is, where do the typical chemicals lie where you have 0 rule of 5 violations, 1 rule of 5 violation, 2 violations, so on and so forth. So in other words, the most drug-like chemicals must lie somewhere, and the least drug-like chemicals must lie somewhere. But where is that somewhere? And you can see it in this plot right here. Again, it's principal component 1 versus principal component 2. And what you find is that this region over here, which was the most dense region there was, and you can see it right there, the purple spot right there, correct? That inset diagram right there, is right here. And you see between 0 and 2 violations of the rules of 5. So the more a chemical looks like a pill, a tablet that you can consume, the more likely that it is going to be present over there. So where do antimicrobials lie? Well, here's a plot where there's principal component 1, there's principal component 2, and these white dots are essentially your antimicrobials. These are FDA-approved antimicrobials. I did have the data with me, so I could overlay that data on top of the complete data set that I had. And you can see that the coverage is actually pretty similar. It's not picture perfect, but it's pretty similar. Another thing that you will notice is most of the antibiotics are right there in the same area where you have the most drug-like substances. Again, this is representing that it is not easy to identify differences between a tablet that you can pop in your mouth and an antimicrobial. But of course, we do know that there are significant differences as well. So clearly, this representation is not going to give you everything that you need. So the next question we asked ourselves was, are there differences between chemicals that penetrate the bacterial cell and chemicals that do not penetrate the bacterial cell? And in all honesty, we found that no, there is not really a huge difference between them. Most of the chemicals are, again, in that spot, and they could be penetrators or non-penetrators. Again, an important thing for us to note is that drug-like chemicals can house both chemicals that penetrate the bacterium and chemicals that do not penetrate the bacterium. We cannot discount chemicals that are drug-like as non-interesting to antimicrobial drug discovery. We looked at it further. For example, we tried to see if gram-negative and gram-positive penetrators might be different. And it seemed like, no, there is not really a significant difference between chemicals that penetrate either type of bacteria. So the next thing we wanted to do is to try and use mathematical methods to do it for us, to identify if there are differences between penetrators and non-penetrators, so that we don't have to resort to crude methods such as just visualizing things in 2 dimensions. So this is what we did. Whenever you try to do modeling of this sort, you're trying to generate a model which is predictive in nature. One of the most important things that you must do is you must do a training set and test set validation. So in other words, whatever models you want to build, you must build them on a separate training set of data. And then once you're happy with your models, you must test it on that validation set right there. It's important that you do so. There are multiple methods that you can use in order to build models. We use these 4: logistical multilinear regression, LMR, linear discriminant analysis, LDA, quadratic discriminant analysis, QDA, and random forest. The first one is a linear method that will help you to classify data into different categories. The second one is a different linear method. It's known as discriminant analysis, but again, all of these are categorization methods. And the third one is a quadratic method. So it doesn't use linear equations, it uses quadratic equations. And finally, random forest is probably one of the simplest learning methods, or what you might even call the very beginning of artificial intelligence in order to build models that will do predictions. Then, after building the models, we went ahead and tested performance. And we did the usual model performance test, which is to look at accuracy, precision, recall, et cetera, et cetera. And of course, we did the statistical analysis to make sure that the probability of seeing better results in one model compared to another, you should not be able to see it purely by chance. So I'm just going to jump directly ahead into the results of these different models. This is the full data that we obtained for all of the different models. So this tells you the model type. So that's random forest. And these are the 3 different linear or quadratic classifiers. And each of these methods was used in order to build a model for all chemicals. So either they penetrate or fail to penetrate both gram positives and gram negatives, or actually either. A separate model was built for gram positives, a separate model for gram negatives, and one separate model for chemicals that will penetrate both. And I seem to be having some sort of my computer. Give me a second. Don't know if I'm still online.
Yes, you are.
I am? Okay, because I can't see anything on my screen.
All right. We can see the PowerPoint.
Sorry, give me a second. Okay, let's hope that this comes back. Okay, PowerPoint was closed. My apologies. Let me bring it right back up.
Okay.
Excuse me for just 1 second. I'll be right with you. Hmm. Okay. Let's get back to full screen mode. I think the computer is trying to tell me that it needs a break.
Yes.
All right, let's see if I can actually quickly get back to the slide that I was on. No, I don't want you to. Oh no. Oh no, not again. Interesting. I've never had this problem in the past 3 years that I've been using this computer. It just had to happen today.
Sorry. So anybody has any questions in between? I think you can unmute yourself and ask during the lecture itself.
Oh yes, please feel free. I like my lectures to be interactive. It's bad enough that we're doing this online. It's better if you ask questions.
Something in the bag.
Sir,
Mm-hmm.
yeah, I have a question. So,
Sure.
this is Shankaran. So,
It does.
especially the talk was very an eye opener. Especially, it was very intriguing to hear that the antimicrobial resistant genes are already present in the microbes.
Oh yes.
Okay, but from what you said, I was able to understand that it is not present in the microbes that are not even exposed to drugs. Am I right?
Not exactly, no. So if it does have the gene, unless there is something that is stopping it from expressing the gene into protein, it definitely is resistant.
Okay. Okay. So it is inherently present in it, but only it is expressed when some antimicrobes is coming, right?
It depends. So
Okay.
there are some genes. which may be present in an inherent manner and are only activated when an antimicrobial is introduced. Some mutations are needed for that. But there are certain other types of genes, such as beta-lactam resistance genes, which have been passed from one pathogen to another.
Yeah. Okay.
Both can happen.
Okay. So my question was why.
Mhm.
So if it is inherently present in the microbes, so
Right.
it when we treat the microbes initially with the antimicrobials, it should
Uh-huh.
actually the antimicrobials are able to mitigate the activity. So, but what, as of what we understand now, it is the microbes that is developing the resistance.
So you're confused about why it has the gene, but it is still susceptible, correct?
Yeah, exactly.
So I'll explain this using one example,
Okay.
right? And it's a very complicated issue. So
Okay.
please take the next few words for what they, with a pinch of salt, basically. There's more details that you need to explore.
Okay.
So for example, um, vancomycin resistance genes, those are transferred from a different pathogen.
Okay.
Uh, and I'm talking about VRSA, vancomycin resistance, which actually causes a change in the target of vancomycin.
Okay.
However, there are certain genes which are inherently present in Staphylococcus aureus, such as the VRAS gene, the VRA gene set,
Okay.
which is inherently present, and it is typically not able to express that protein quite as much.
Oh,
But when you use vancomycin against Staph aureus,
okay.
only the subset of Staph aureus which is able to express that gene more will survive.
Okay.
And then you get resistance. Does that make
Yeah.
sense?
Yeah. Exactly. I understood. I understood. Thank you.
There you go.
Okay.
Okay, I think I'm back. I'm going to try and share my screen again. Here we go.
Okay, thank you.
Share my screen. I really hope this works because I'm only halfway done. Okay, let's try that. It's visible?
Yes, it is.
Excellent. Okay, I'm going to try and shift you over here again. All right, so let's try this again. So let's take a look at model performance. So again, for each different type of bacterium, we actually have a separate model built using all of these different techniques, right? So the first thing that I looked at was the phi value, which is also known as the Matthews correlation coefficient. The closer you are to the value of 1, the better your model is. It's literally an indicator that what you are seeing, the predictive nature that you're seeing, is not happening purely at random or purely by chance. So the only case in which you had truly good predictions was in the case of random forest. So I went ahead and discarded all of the other all of the other models, and just looked at random forest next. So what you see is that the overall accuracy across the different types of pathogen is relatively high. It's somewhere between 80% and 92%. What you also see is that the recall is pretty high, over 90%, actually close to 100%, with the exception of gram-negative recall. So recall is basically, if I want to express this in simple words, if you are to say, if your model is to predict something is a penetrator, what percentage of the time are you correctly making that prediction? So clearly, virtually 100% time in 3 cases, but in the case of gram-negative pathogens, it's only at about 65 to 70%. Why is that the case? an analysis of the data set tells us that the total number of data points for chemicals which penetrate or fail to penetrate the gram-negative pathogens is much less compared to the data on chemicals that penetrate gram-positive pathogens. So because of that lack of data on what will penetrate gram-negative pathogens, you have a very low recall value in comparison to the rest of the models. That makes sense. So let's take a look at the negative predictive value and specificity. So specificity was a major problem in most cases, except in gram-negative. So in simple English, if we are trying to demonstrate that, or if you're trying to make the model predict that a chemical is going to be a non-penetrator of bacterial cells, what percentage of time are we correctly making that prediction? So in all of the other cases, gram-positives, both gram-negatives and gram-positives, or either, we're making between 40 to about 60% correct predictions. However, in the case of gram-negatives, we're making it correctly at 91%. And the reason for that is because we had more data for gram-negatives on chemicals that do not penetrate. But in my opinion, more importantly, look at the negative predictive value. It's consistently between 80 to 90%. What this means is that if our models are saying that something is not going to penetrate a bacterial cell, it is going to make that prediction correctly. So overall, at the end of this paper, there's a lot more to the paper. I'm not getting into all of those details, but the question we have to ask ourselves is, have we actually solved the problem of chemical space? No, but we have made some progress in that regard. So we have demonstrated that if we are given appropriate data, and mind you, we don't have picture perfect data yet. We're still trying to gather good data, right? But if we have good data, then statistical analysis will potentially help us. That is something we have demonstrated reliably. There are plenty of weaknesses that we need to overcome, right? I already stated something about the dirty data set. It's a problem. We need clean data and we don't have it. We're still categorical data. We're dealing with categorical data. We need to get continuous data. That's actually going to be better for model building. We know for a fact that the random forest models are memorizing a little bit. So it's taking this data point and it's saying, okay, it seems like it's a penetrator. So if this data point is right next to this one, this has got to be a penetrator as well. It's more of memory than really a mathematical model, if that makes sense. But that's not necessarily a bad thing. I mean, it's at least giving us reliable predictions, right? As a starting point, that's not bad at all. I talked about the categorical data already. We need continuous data. So we need to measure, for example, the kinetics of permeability or something of that sort, and use that as an endpoint rather than just categories of what gets inside and what does not get inside. Also, we are at this present time not looking at specific species. We're not looking at strain-dependent differences. So we just mentioned Staphylococcus aureus a few minutes ago, right? There are MRSA strains, there are visa strains. Even within MRSA, there are so many different substrains that are very well known and documented. I'm fairly certain that if we start looking at patient samples, we'll find even more numbers of strains. So what are the differences between these strains and how do they dictate the ability of chemicals to get inside the bacteria? There are so many different things that we need to explore and we have absolutely no idea about. So those are all things that we need to focus on. And clearly we need to look at chemical nature itself. Right now, we're just looking at hydrophobicity, size, common descriptors, but they don't seem to be giving us a difference between penetrators and non-penetrators. We can't continue to focus on the same definitions of chemical properties. We have to look at other properties as well, and we have no idea what those properties are. Okay, that's enough about finding chemicals to run our screens. Let's take a look at how to design a decent virtual screen. And I'm going to do this in two parts. So basically, two questions that we need to answer are, do we really need to run a full docking and scoring run? Docking and scoring is probably the most commonly used form of virtual screening. Do we really need to run a full-fledged docking and scoring? And if you do have to use docking, how do you know that these theoretical calculations actually reflect reality? Those are two questions that we absolutely need to answer. And I'm going to demonstrate a couple of things to you using two pieces of my own work. One of them is drug discovery against RMLA from Pseudomonas aeruginosa. And the other one is more recent and unpublished work on STK1 from Staphylococcus aureus. So the RMLA work was published right over here. So let's talk about RMLA. The goal was to identify chemicals that inhibit a protein called RMLA, which is a glycosyl transferase. It is required for Pseudomonas aeruginosa to survive inside a host. So my collaborators at St. Andrews came to me saying, hey, listen, we ran a 15,000 chemical high throughput screen, and we came up with 2 hits against purified RMLA. Both of them, we have crystallized both of these. If you know Dr. Venkat Subramaniam's background, he's done a lot of crystallography at exactly this same university, University of Dundee. They're very good at it, right? So the first thing they did was they did crystallography, and they brought me the crystal structures and the chemicals and said, find me some inhibitors. We also had a crystal structure for the natural inhibitor of the same protein. So this is all the data I had. I had to find them some new inhibitors. The first thing that most people would do is, well, let's run some docking and scoring. Okay, you could do docking and scoring. That's a theoretical method, and you basically spend a lot of time optimizing the docking and scoring process. Then you ultimately end up with, if you're lucky, a 30 to 40% success rate. And there you go. Well, I did something a little bit simpler. I went ahead with a ligand-based screen. So what I did was I said, well, it looks like this scaffold is common in between the 2 hits. And also, this looks extremely similar to the thymidine of TMP, which we know binds at that same location. So why not just look for chemicals that are similar to it? Guess what? That simple screen on the Zinc database, I don't know if you know about the Zinc database or not, but it's a publicly available database which is maintained by UCSF, University of California at San Francisco. Most of the chemical vendors always submit their chemicals to Zinc because most people in the entire world can actually run their searches on that database and find chemicals from multiple vendors, and they can purchase them from the vendors and test it in-house. And it's got some pretty nice tools in order to do that kind of work. And you can see the number of chemicals that have been submitted to it. It's actually close to 1 billion, of which roughly about 250 million chemicals are purchasable. Well, with that kind of numbers, it was easy for me to find a good 50 chemicals that looked kind of similar to this. And I ordered them and I tested them. And you know what? I ended up with an 80% success rate. Docking and scoring, few weeks of work, 20 to 30%, maybe 40% success rate. Being smart, running a ligand-based screen, half a day's work, 80% success rate. You tell me which one you would pick, right? We got a bunch of hits right here. Many of them are reported. These are not all hits directly from my screen. There are actually a few chemicals which I designed based on the initial couple of hits. One of the graduate students at St. Andrews actually synthesized a bunch of these chemicals. All of the synthesis is reported over there. But beautiful work. We ended up getting a bunch of crystal structures again, because like I said, they're really good at crystallography, right? So they went ahead and crystallized all of them. Ooh, excuse me. And we have hits. So again, let me ask you, was it necessary to conduct docking and scoring? Absolutely not. When you have a smart opportunity to expand the number of hits you have for a relatively low price, do not get into complicated things which might give you worse off results compared to what you can do in simple terms and get better results. Always do the smart thing. Okay, so let's assume that you do have to go ahead and run a docking and scoring screen, right? You've got to validate your methods. And that's what I'm going to show you using the example of STK1. And for some reason, did I just run out of battery? Oh, no, not really. Oh. Excuse me. All right. I'm just going to use my mouse. So if you do want to do a docking study, then you need to validate your methods. And I'm going to show you exactly how to do that over here. So STK1, which is also known as PKMB, is a global metabolic regulator in Staphylococcus aureus. It is a serine threonine kinase, very similar to the serine threonine kinases that you can find in our own body. It has been demonstrated that if you knock out the STK1 gene, then the resulting strains of Staphylococcus aureus have a reduction in the MIC of beta-lactam. So in other words, you need less beta-lactam antibiotics in order to kill those versions of Staph aureus. So in other words, if you can inhibit STK1, what's going on here? Right. If you are able to inhibit STK1 using chemicals, those chemicals could be useful as therapeutics in conjunction with penicillin G as an adjuvant. So let's try and optimize a docking protocol for STK1. What do we have? We have a crystal structure in the PDB, 4EQM. I have given you the code right here. So you take this structure, you prepare it, and you run a docking study. In order to do that, first of all, you have to identify the appropriate software and settings that will do a proper docking. So what I did was I took the adenine part right here from the co-crystallized ligand, which is AMP-PNP, a non-hydrolyzable ATP analog, and I redocked it into the crystal structure. I went through a couple of different types of software. I ended up with Plants, which is from the University of Tübingen, from Thomas Exner's group, and ChemPLP. These work perfectly fine out of the box without any issues. Another thing I did was I validated it. How did I do that? I took 14 known inhibitors and mixed it with 10,000 decoys, which are from a kinase focused library. So they look like kinase inhibitors. I docked all of them in a two-step process. The first step was that I with all of these computer issues. Okay, so I docked them in, and I identified those chemicals which were docking into the ATP site with an RMSD of less than 1. In other words, in triplicate, I was able to demonstrate that those chemicals would be docked in reproducibly. That's step 1. Step 2, I use a pharmacophore-based screen and identified chemicals which form very similar hydrogen bonds when compared to AMPPNP. So if it's docking reproducibly in the same binding pocket, forming the same bonds as AMPPNP does, I hypothesize that it's going to be a true inhibitor. And guess what? 12 of the 14 inhibitors passed the test. Only less than 1,500 of the 10,000 decoys passed the test. The probability that the known inhibitors would be enriched to this level purely at random, by chance, by accident, was less than 1 in 10,000. This validated my docking protocol, and I went ahead and essentially I used this docking protocol in order to dock a known kinase focus library into STK1. I essentially ended up with a bunch of chemicals that looked like they might be good inhibitors, of which this one was of particular interest to me. We're calling this 3M09. I won't get into the reasons of it, it's just procedural. 3M09 seems to form virtually the same interactions with the backbone of STK1 as does AMPPNP. I went ahead and tested it in-house. A protein thermal shift assay basically told me that it definitely stabilizes the STK1 structure thermally. So that tells me it's confirmed it is an inhibitor. And against the pathogen, I was able to demonstrate that it reduces the MIC of MRSA from over 256 micrograms per ml to about 64 micrograms per ml, which is an 8-fold reduction. Visa, it does from 32 to 8. That's a 4-fold reduction. It also reduces the MIC of vancomycin for Visa from 8 to 2, which makes it, so Visa is known as resistant because its MIC is over 2. So it's above 4 or above, right? It reduces it to the susceptible zone of vancomycin, which is a really tremendous find. So clearly this chemical is of interest. We are trying to work more. All of this information is confidential, by the way, so please keep it to yourself. Although I know it's streaming on YouTube, so I don't know why I said that. Okay, anywho, so confirmed, right? Experimentally, we have demonstrated that the protocol works. So let's summarize this part. How do you design a good screen? So try to avoid using theoretical methods when you don't have any validation in place. A good ligand-based screen will likely help you generate a lot of data really quickly, which a really complicated other theoretical method is going to be difficult to justify. Just don't do it, right? And if you do have to use docking and scoring, make sure that you validate your process. We see a lot of documents, manuscripts for review, where people claim that the findings from docking are as good as experiment. They are not. You have to validate, you have to demonstrate that it actually works. Then and only then will it be accepted as reality? And then if you are able to demonstrate in your lab, wet lab, real experiment, that it works, that's when you know that it works. Okay, the last one, target validation. I'm going to do this quickly, and I know I'm running a bit behind because of various issues. So the question we are asking ourselves is, what do we screen against? Okay, we're going to do a virtual screen. We've accepted that we can do ligand-based screens or docking and scoring screens, maybe a few other things also. But what do we screen against? That's the other question you need to answer. So this is summarized really nicely in the druggability problem. So again, there are a lot of different drug targets, many of which are biologically validated. In other words, they have key roles in biological processes, but chemicals do not bind them very easily. In other words, you pop a pill, it reaches the drug target, but it doesn't do anything. Useless, right? So there are some good examples. Factor 10A used to be recognized as a less druggable target. You need a very specific pharmacophore in order to achieve high affinity against that particular enzyme. Compare that to certain kinases such as BCR, ABL, you'll find kinase inhibitors like that, right? So that's much more druggable compared to factor 10A. So this is well known. And it has to be, the secret to why something binds well has to be in the binding pocket. So we're looking at the binding pocket, not the whole protein. So let's go back to STK1 because this was a really interesting case. You literally have an orthosteric pocket and an allosteric pocket. These are the active sites, the orthosteric sites, and these are the allosteric sites. Both of them bind AMP. I'm sorry, that's not supposed to be AMP, that's supposed to be TMP. Both of them bind TMP, right? Both of them bind TMP. So the question is, which binding site should be druggable? If you're able to demonstrate that experimentally, that's brilliant. So theoretically, how do you find druggable pockets and how do you segregate them from the less druggable pockets? That's the big question. And here's a paper that we published in the early 2010s. We came up with a large database, just like in the case of chemicals that penetrate versus chemicals that don't penetrate. In this case, we were trying to find pockets that will bind or will not bind drug-like molecules really strongly, right? So we needed a large database. We had at that time what was the largest set of pockets where we had the definitions and it was well accepted. We went ahead and built models using that data set to try and identify or predict which ones would be druggable, which ones would not be druggable. These statistical models were based on clearly the property descriptions. And we wanted to check whether or not these models that we were building, these statistical models, do better than random allotment. And then we use external data sets in order to demonstrate that the method actually works. So I'm just giving you an overview because I know I'm out of time. So here's the general process, okay? So here's your protein. You identify the pocket. You identify the properties of that pocket. You do this for a whole database of pockets. Then you build models, and then you try to validate it. So initially, we took all of these different properties, we calculated them for each pocket. We went ahead and we built the statistical models, and then we validated it using a test set. So here's the training set data. What you see is in dark are the druggable pockets. In light gray are the less druggable pockets. Can you see that there is a separation between them? That means that the model is working internally. It's a mathematical model that is making correct predictions. But This is just the training set. We went ahead and did a test set performance as well, right here. So the model that was built on the training set had never seen the test set, was used on the test set to make predictions. And again, you see a very clear separation of the druggable pockets in dark from the non-druggable pockets in light. The model works. We went ahead and did a lot of other, it's going to crash again, I think. Wonderful. Ha, it's midnight. Okay. I'm going to come back to Zoom real quick. Dr. Venkat Subramaniam.
Yes, sir.
I think I'll probably finish this presentation without my PowerPoint slides. I have no idea what's going on, but it's really giving me some trouble.
It's quite unusual. It's quite unusual. Yeah, I have never had a problem with mine neither. So
I don't think it's Zoom. I think it's my computer. It's basically decided that it's too tired to continue, and it's telling me enough is enough.
Sure, sir.
So my sincerest apologies to everybody. But I think I may have by now demonstrated that this model is able to identify chemicals which will bind a binding site really strongly. Do you agree with that? Anybody? Say yes, please.
Yes.
Okay, good. All right. So what I have tried to show you today is that it is not just enough to go ahead and run a virtual screen. There are three things that you absolutely must think of. Number one, what chemicals are you going to work with? It's not necessary that you have to identify chemicals the way I have, because my problem was specific to antimicrobial resistance. However, I have worked with bacteria. The same holds true for multiple types of systems, fungi, nematodes, infections in general. Chemical space definitions of what chemicals are useful is very difficult. Two, when you run a virtual screen, be smart about it, right? If you don't have to use complicated methods, don't. If you do have to use complicated methods, validate them. Make sure that you have strong validation to demonstrate that you have real results. If possible, experimentally demonstrate that it works. The third thing is, don't just go for any drug target. There are plenty of drug targets that will not stick to drug-like chemicals. You don't want to work with those chemicals, those targets, okay? By the way, the last part of that was I would go back to RMLA, which was my Pseudomonas aeruginosa protein, and I would demonstrate that my method predicted the allosteric site to be the binding site for most of the chemicals. And crystallographically, we have demonstrated that that is exactly the site that it goes and binds to. So if you have strong methods of this sort, your publications will be in very good journals, and it'll really help. And I'll end by saying, we've only taken some small steps towards solving the problem of antimicrobial resistance. And let's just say that we have climbed up on a molehill and we need to climb Mount Everest. I hope that makes sense. And if you have any questions, please feel free to ask away.
Thank you, Arijit. That's a very interesting talk. So I myself learned a lot. Looking at things at a different perspective, because we know that there is antimicrobial resistance is it's an important problem, but there is so much behind it. Is something that we are not aware of. That's a good insightful talk, actually. I do have just one question, because we talked about a validation of molecular docking or screening, and you carried out certain validation of the docking procedure. But does that procedure has to be carried out for every different target? Because how specific is that validation? So is it just, for example, you have carried out for one target, but can you just extend it to other targets, or is it better that I do it for every different?
Validation has to be carried out for every single pocket, even not just target, but also pocket. It becomes difficult sometimes. Sometimes you just don't have the amount of information necessary to really seriously carry out a proper validation. In that case, sometimes you have to resort to trial and error. So, okay, let's identify 50 chemicals. Let's see how many of them actually end up working. But there are some common sense things that you can do. Number one, you can definitely make sure that multiple docking runs are giving you the same results, right? Because without reproducibility, what use is a simulation? Then let's say that there are some hydrogen bonds that seem to be really important in a biological sense. Are those hydrogen bonds being, or are those groups of the protein being engaged by the ligand that has reproducibly docked? That's another very common sense thing you can do and say, hey, this looks like it might be a likely inhibitor. So some common sense things will help you get some initial results, and that will probably help you with a lot more afterwards. That's the way I would do it.
Okay, thank you. So another quick question based on your last statement is, but you said that you identified that allosteric binding pocket is better than the extent. But is that, can I extend that to all proteins?
So I'll have to admit that I love to use that in my presentations, that I was really successful, but that's an N of 1. Right? The method definitely worked on everything we had internally. So the analysis was not prospective, it was retrospective. The only prospective case in which we were able to apply the method was RMLA, and it proved to be successful. And then unfortunately, we ran out of funding for the Aeropath project and we couldn't test any further.
Sure, I understand. Okay, thank you very much. So does the participants have any questions?
Sir, I have a question. So you were suggesting us to do ligand-based screening instead of docking, right? So did you feel, are there any trade-offs in that method? So usually people go with docking, but you suggested to do ligand-based screening, and of course it has happened successfully. So are there any trade-offs?
There's a major trade-off. Docking roughly about a few million chemicals into your structure gives you a lot of structural variability in your inhibitors. That won't be possible with ligand-based screening.
Yeah.
However, you'll get your results faster and more reliably, and you'll be able to put together a nice publication and preliminary data for a grant.
Okay.
That's the trade-off.
Okay. Thank you, sir. Thank you.
No problem.
Any other questions? Okay, following up on that question, so I may have another question.
Sure.
So what, because the ligand-based screen gives better results, but then you're restricted to just one or two scaffolds that you can look for. But what if we do a screening and then identify potential scaffolds and then do a ligand-based screen based on that? Would that be?
Yeah, that's exactly what I'm doing right now in one of my projects. So I have some data that I've generated in-house using a ligand-based screen. I'm putting it together in a grant, and I'm saying, here are further ligand-based screens that I'm going to do where the scaffold is actually different. to see whether these other scaffolds, which are close but not exactly the same scaffold, are going to work. So computational chemistry is all about cleverness. When you don't have data, how clever can you be?
Yes.
And how effective can you be? That's it. That's what it's about.
Okay. Thank you. Anyone else have any questions? I think I will stop here. So once again, I will thank you for staying so late.
No problem.
Giving us a talk, which was very interesting, of course, I must admit. And thank you very much, sir.
I'm glad you liked it. It was an honor to be invited. Thank you so much.
You're welcome, sir. Thank you.
I'll log off now, right?
Yeah, we can talk.
Okay. Thank you. Nice meeting everybody. Have a good night or good day. Sorry.
It's morning. So this first session has got over, so we have 3 more sessions.
Oh, okay. Bye.
Okay, thank you. Participants, and then we'll take a break. We'll be back on 11 o'clock, where we'll be having a hands-on session on high throughput screening using Pyrex, which will be given by Dr. Ramya. Please log in remotely using your Windows ID. Let's report back. So before that, I would also say that we will circulate a Google form that will requesting a feedback on the sessions so far. So please take a few minutes to fill that Google form also. So I'll communicate it by WhatsApp as well as email later today. So please do fill them. Thank you. So we'll break now, and then we'll be back again at 11 o'clock. Thank you.